acidic and basic nature of your different organic compounds. We're going to be using your Broadside-Lowry definition for an acid and a base. So an acid is a proton donor and a base is a proton acceptor. They're going to have two factors that influence the acidic and the basic nature. These two factors are your inductive and your conjugate effect. So in terms of your inductive effect, it can be broken up into your negative inductive effect and your positive inductive effect. The stronger the negative inductive effect, the stronger your acid. The stronger the positive inductive effect, the stronger your base. Under your positive inductive effect, you're looking at electron pushing groups. So groups such as your methyl are pushing electron density from itself onto your carbon within oxyl ion group. Under negative inductive effect, you're looking at electron withdrawing groups. So they're doing the opposite, whereby they are pulling electron density away from the attached atoms towards itself. Now we're going to be covering or recapping your conjugate/your resonance. This occurs in compounds where you have that alternating double and single bond. It comes from the overlapping of your pi bonds, and this is going to cause your electrons to be delocalized. And this delocalization also increases the stability of your structure. So in terms of your carboxylate ion here in your acid, after the loss of your proton, you end up with one oxygen carrying 3 bonds with a negative charge and the other oxygen carrying 2, so that's a neutral. So this stability causes your carboxylate ion to be relatively stable, and so the ability of it to recombine with a proton or H plus is reduced because it's not as negative and therefore is not as attracted to your positively charged proton. We're comparing the acidities, so when you're looking at your carboxylic acids, in this example it is your ethanoic acid, your phenol, and your ethanol. So we're going to start with the one with the lowest pKa. Again, the smallest pKa lets you know that that's your strongest acid. So in terms of your ethanoic acid, this is exhibiting both your positive inductive effect and your resonance. Once that proton, that H plus, is lost and that bond is broken, you now end up with a alternating double and single bond and this carboxylate ion here and this particular region of your ethanoic acid. Though that alternating single and double bond here actually results in your delocalization of electrons because a lone pair from your oxygen is actually close enough to overlap with that carbon there. Alright, so on this side you have your resonance stability. In addition to this, there's another factor, which is your positive inductive effect caused by your methyl group. This methyl group here is pushing electron density onto your carboxylate ion region, making it a bit more negative. Okay. So even though it's a bit more negative, that might cause it to pick back up the proton. Your resonance stability is also there, and that is a stronger factor. Next, we're looking at your phenol. So in the case of your phenol, you're going to have the same H plus of your proton leaving your phenol once that bond is broken. In addition to this, your phenol has the conjugate or the resonance effect. So after the loss of the proton, it exhibits resonance stability. So that is because one of the lone pair of oxygen is overlapping with your benzene ring, which already has resonance stability. So this overlapping of the lone pair on your oxygen with your benzene ring causes your delocalization of electrons to spread from your ring to your oxygen. That's giving your phenoxide ion that gets formed more stability, so it's less likely to pick back up a proton. Your last example is your ethanol, and that is an alcohol. So in this case, you don't have your resonance effects. So after the loss of that H plus there, you just have a very strong negative charge concentrated on that oxygen there. That very strong negative charge is going to easily attract back a proton and easily form back a bond with your H plus. So that's why your ethanol is a very, very weak acid. So for your ethanoic acid, the different conical forms are actually equally distributed. So regardless of which position double bond takes, it's the same adenoid ions that are being formed, and they would have the same degree of negativity. Your negative charge of oxygen is now equally distributed between the two oxygens. In terms of your phenoxide ion, you do have the delocalization or the resonance between your oxygen and your ring, but the position of your double bond are not equal. Alright, so these result in resonance structures are not equally contributing. So it's less stable. So some of your conical forms are less stable than others. So that's why your ethanoic acids or carboxylic acids are stronger than your phenol. So next I'm just recapping your basicity. Alright, so again you're going to have that conjugate or that resonance effect, and you're also going to have the inductive effect. But in terms of your basicity, your positive inductive effects is going to make your bases stronger. Here we have ammonia, methylamine, and your phenylamine. So again, your pKa range is the same as your acids. The smaller the pKb, the stronger the base. In terms of your bases, they must have a lone pair of electrons in order to accept a proton or pick up a proton instead of donating, because we're looking at bases now. So comparing your ammonia, your methylamine, and your phenylamine, they all have that lone pair of electrons, allowing it to pick up a proton. So as enough with ammonia, all it has is that lone pair available to pick up a proton. Right. Next is your methylamine. It has the lone pair of electrons to pick up a proton, and it also has a methyl group. And this methyl group is giving it a positive inductive effect, meaning that this methyl group here is pushing electron density onto your nitrogen region, making it more negative and more likely to attract a proton. So it's easier for your methylamine to pick up a proton and form a bond with your H, utilizing its lone pairs of electrons. Your last example is your phenylamine. Now, in terms of your phenylamine, there is a lone pair attached to your amine group or your NH2 group. However, similar to your phenol and your phenoxide ions, that lone pair of nitrogen actually overlaps with the delocalized ring of your benzene. And this overlapping causes your delocalization to spread out to your nitrogen and within your ring. Your lone pair is now overlapping with your ring. It is no longer available to pick up a proton or pick up an H plus ion, making it a very, very poor base. It's going to require a lot of energy to break that delocalization just to make that lone pair available to pick up a proton. So it's very, very unlikely that your phenylamine is going to have that lone pair easily and readily available to pick up a proton. Bases, their positive inductive effect increases the basicity of your bases. Right? So that means the more electron donating groups present, the more positive the inductive effect and the stronger the base, because they're increasing the electron density and therefore increasing the negative charge on your nitrogen atom, making it more attracted to your H plus or your proton. So now with that recap, we have a few questions that you're going to be doing on your own. I'm going to look at them in class. So lastly, we're going to wrap up this topic by looking at your amino acids. So your amino acids are simple organic compounds, and they're the building blocks for your proteins, aiding the formation of other compounds. Alright? It's important to note that your amino acids are actually amphoteric and can act as buffers. So in this image here, you have the general structure of your amino acids. You have your amino group, you have your carboxyl group, and then you have your hydrogen, and then you have your R group, which varies depending on which amino acid you're looking at. The nature of your amino acids makes it polar and unpolar and so on and so forth. So here are some general examples of amino acids. The simplest amino acid is your glycine, and that's just with your amino group, your carboxyl group, your H, and another H. So all of your amino acids are going to have that. In addition to this, your amino acids actually exhibit chirality. It is just referencing your carbon here that is attached to 4 other different groups, with the exception of your glycine. So you have covered this under your different types of isomers, where we're looking at your optical isomerism. And these are also known as your enantiomers, meaning that they cannot be superimposed on each other. So similar to your hands, they're mirror images of each other, but they can't be superimposed. Amino acids are neutral species, and it's gonna exhibit a positive and negative end, right? So that positive and negative cancel out each other, making it neutral. That positive side comes from your amino group, and the negative side comes from your carboxyl group. So depending on what solution your amino acid is in, it can either act as a base or act as an acid. So here we have the acid to base nature of your ions. So in a neutral solution, both your NH2 and your COO groups are ionized. Okay, so they're now your NH3, and now you have your C, double bond O, another O with a negative charge. In neutral solutions, these are ionized, meaning your positive and negative charges cancel out each other. In an acidic solution, your amino acids are going to be taking up protons from that solution. So these with the ions, your NH3 is already protonated, so your C double bond O is acting as your proton acceptor. It's negatively charged, going to pick up that H plus from your solutions, right? Versus in an alkaline solution, it now has to donate a proton to the solution. So in this case, your NH3 plus is now losing a proton to give it to the solution. So this ability to pick up or donate a proton causes your amino acids to resist small changes in pH. This is why amino acids can act as buffer solutions. So because they show both acidic and basic properties in solution, your C double bond H group reacts with your bases to form a metal salt. So acid and base react together to give you your salt and your water. Same thing's happening here. So your NH2 group will react with your acid to form your salt,